I often wonder whether this huge variability, this complexity, is not necessarily linked to a unique function, but rather there is a very high level of redundancy in the brain that, was never punished, evolutionary speaking. All these aspects have to do with efficiency in terms of computations and utilization of resources. And I think that's where really the dendrites shine. Initially, I was thinking that dendrites are really, really important because they allow the brain to do more advanced, let's say, computations that we could do. But I now kind of think that their more important role.
This is Brain Inspired. I'm Paul. Panayiota Poirazi is my guest today. Yiota runs the Poirazi Lab at the 4th Institute of Molecular Biology and Biotechnology. And Yiota loves dendrites, those branching tree-like structures sticking out of all of your neurons. And she thinks that you should love dendrites too, whether you study biological or artificial intelligence. So in neuroscience, the old story was that dendrites just reach out and collect incoming signals for the all-important neuron cell body to process. Yiota and people like Matthew Larkum, with whom I chatted in episode 138, are continuing to demonstrate that dendrites are themselves computationally complex and powerful, doing many varieties of important signal transformation before signals reach the cell body. So for example, way back in 2003, Yiota showed that because of dendrites, a single neuron can act as a 2-layer artificial neural network. And since then, others have shown that single neurons can act as deeper and deeper multi-layer networks. On top of that, and in Yiota's opinion, an even more important function of dendrites is increased efficiency in computing that they bring to the table, something that evolution surely favors, and something that artificial networks will need to favor as well moving forward. So we talk about some of her work and reflections and ideas about the computational properties of dendrites with respect to cognition and efficiency. As always, go to braininspired.co if you have a couple bucks to support this podcast through Patreon. Or if you're interested in learning more about neuro-AI topics, you can check out my online course, Neuro-AI, The Quest to Explain Intelligence, also at braininspired.co. Show notes for this episode are at braininspired.co/podcast/167. Okay, here's Yiota. Yiota, you've been studying dendrites since your master's thesis, at least. So I was kind of assuming that maybe you came in from studying something else and eventually came to love dendrites. But have you always been interested in dendrites, or how did you come to love the dendrite?
Good question. Well, I was always interested in the brain, right? Since I was a high school student, I wanted to study something related to the brain. But, this is a long story. There was no university in my home country in Cyprus where they would offer a neuroscience or even a medical degree, let's say, as an undergrad. So I studied mathematics instead. And then I wanted to find my way somehow back into biology and in particular the brain and neuroscience. So I went to USC to study biomedical engineering, which was the way to get back through a mathematics degree. And there I met Bartlett Mel. And I met Bartlett the first few months that I arrived at USC. And he was doing dendrites. So I wasn't in love with dendrites at that time. I just fell in love with them when I started working with Bartlett. So that's how it all started.
So you were at USC and you decided to leave the United States and go back to Greece. And I understand that you were like the youngest research director at your institution. Is that true?
I was the youngest researcher to be hired when I was first hired. So I was 27, I think, when I got my position.
There were only like 3 other people, right, in that kind of area of study. Right?
In the entire country, you mean?
Oh, okay. That's quite larger then. Okay.
No, my institute is pretty big, but computational neuroscience is not big in Greece, unfortunately. There are less than 5 labs throughout the country that do computational neuroscience. So it's a kind of, unique skill around this place.
What about experimental neuroscience? Because I know that you've transitioned your lab, and I'm going to ask you about that later, to include experimentation. Is that a big scene in Greece?
Well, there are quite a lot of people doing molecular neurobiology, but not really systems neuroscience. There are a few people that used to work on monkeys in Crete. Well, some of them still do, but there is not a lot of mice electrophysiology or in vivo behavior. So it's still kind of lonely here. There is at least one other lab that has recently joined and is a person who came from the US and has the expertise for systems neuroscience in mice. And that's why I also decided to expand. So we're sharing a lab, and he's helping a lot with, training and the people in my lab to start doing experiments.
Okay. You said it's lonely. I was thinking that there are pros and cons, right? Because a pro is that you sort of stand out and you don't have much fellow competition in your area. But a con is that it's a lonely endeavor.
Yes. I mean, I think it's really important to have colleagues with whom you can discuss problems, troubleshoot, write joint grants, write papers together, mostly run by them your ideas, right? And I really felt this difference when I was doing my sabbatical at UCLA in 2008. Then it really stood out how different it is to be in Greece and to be, kind of a loner on this endeavor. I appreciated a lot the collegiality that I found there and the opportunity to talk to people on a daily basis.
Okay, so it's like passing people in the halls and actually physically attending talks and having conversations after and before.
Yeah.
All right, so I'm going to kind of skip ahead here because a lot of what you do and have done in the past is computational modeling of dendrites, and you argue essentially that artificial networks need dendrites. And yet, there's been this recent explosion in the capabilities of artificial networks. And I'm kind of curious how you have viewed that explosion and watching artificial networks getting better and better and better. Have you just been thinking, if they only had dendrites the whole time? Or are you thinking, maybe they don't need dendrites? What's that been like?
Yeah. So that's a very important question because I think that brains need dendrites. And there's a big distinction between a brain and an artificial system. And the reason why there is such a big distinction is that the brain is under evolutionary pressure. We have a limited size of the skull. We have a neural tissue doing the computations. We run on very low energy. All these aspects have to do with efficiency in terms of computations and utilization of resources. And I think that's where really the dendrites shine because they bring this efficiency to biological brains. In many ways, we can discuss that. Now, you can think of ChatGPT, for example, right? It's a really big thing right now. It needs the energy of a small city to be trained. It's not comparable to my brain or what your brain is doing. Not to mention the size of the infrastructure, the number of GPUs that are needed to run this thing, right? And it's still not smarter than a 2 or let's say 7-year-old child. So I still think dendrites are really important. They're important for 2 reasons. One, which we haven't really mastered yet in artificial intelligence systems, is like the ability of the brain to learn continuously, lifelong learning, for example, or to transfer skills from one task to another. There's great improvement in the new systems out there, but this is still an open task, I think. And the most important aspect, obviously, is efficiency, sustainability. I mean, we are reaching a ceiling in the technology of chip manufacturing. And this is not enough to answer to the very high demand of computational intelligence systems that we need on our daily lives. So efficiency is going to become a bottleneck in the next few years. And there are already a lot of people out there talking about neuromorphic computing with the idea of doing what the brain is doing so that we can achieve the same or better, let's say, computations and intelligence with a much more sustainable technology, smaller, cheaper, low cost, low energy. And I think that's where dendrites really matter.
So I appreciate the efficiency story and importance of efficiency, but there's a part of me that is more excited about the expansion of cognitive abilities, the improvement in cognition for dendrites, because we're living in a world today, right, where yes, it costs a small city to train these models, but you can just go to a medium-sized city and it gets smarter, and a bigger city and it gets smart. Maybe eventually we'll get to the Earth-sized models and it'll just be like super smart. So do you think that do you think that scaling up will just accomplish the same cognitive capabilities, or is there some secret sauce in dendrites?
That's a wonderful question. If you had asked Matthew Larkum, I'm sure he would say that there is a secret sauce there. I am not entirely convinced, to be honest, because deep neural networks, which is essentially, the technology behind these systems, right? So if they scale big enough, they should be able to approach any problem, any kind of computation. At least that's what we know from theoretical math, that they're general approximators. They should be able to solve any problem, right? So it could be that we haven't really reached the size of the network that is required to come up with cognition. We don't know, right? It is also possible that there is something special about dendrites, which, however, we have not really put our finger on yet. I mean, there are really cool things that happen in dendrites. And one of the really interesting things that our work with Matthew, for example, is looking at is related to perception. And Matthew, these are not my studies, so I'm not taking any credit for this. So Matthew has shown that when the animal is very close to perceiving a new sensory stimulus, that is when the dendrites light up. And you have this massive activity measured with calcium imaging in the tufts of the distal dendrite of cortical neurons when the animal perceives a stimulus. So it is possible that without dendrites, you cannot have this aha moment, right? Which is what happens when you realize that you're now perceiving something. So that's the other possibility that answers your question with respect to the magic sauce. Can we not do that with dendrites? Is it really that we need dendrites to actually have this kind of computation? I don't think so, because in terms of a computation, what happens is now the dendrite is signaling to the cell body that something really important is coming in. So, kind of pay attention to it. And it's done within the single neuron by the dendrite talking to the cell body, which otherwise would not have done, right? If it's a not important signal, you would not see these calcium, these dendritic spikes. You could definitely do that with a pair of neurons, and you have them disconnected, and then they talk to one another under specific conditions. Now, the brain might want to do this with dendrites, again, for reasons of efficiency. You save much more space by not having, these cell bodies that are huge in the medium and having them talk to each other conditionally. You also have a lot of dendrites that cover a very small surface area so that you can have a lot of combinations of when you want this dendrite to talk to the cell body. And therefore, presumably, you can think that you can, let's say, learn many different things by combining the different dendrites with the same neuron, right? Which if you did not have dendrites, you would have to have as many neurons to implement this computation. So I'm not sure whether it is a matter of a special sauce or just a matter of, this is a much more efficient and smarter way to do this, to solve the same problem. We don't know for sure. I mean, I wouldn't bet all my money on the fact that there is no special sauce in dendrites. In fact, I wish there is, right? I've dedicated my life studying on dendrites. We just haven't seen it yet.
I was thinking, I just had the thought that a line in math, right, is an infinite set of points all jammed together. And I mean, not only would you consider, replace a dendrite in an artificial system with another unit, but you might have 100 units all connected together, because dendrites are so complex. They have these propagating properties, and they have different shapes. They have different ionic conductances, and all sorts of things are happening and in constant flux. And so is that a crazy idea to just chain up a bunch of units together with different properties?
It's not a crazy idea. And people have already proposed this chain of units that represent, let's say, dendritic compartments, not the whole dendrite, each of which is doing its own computation. And this is supported by experimental data. In fact, we have proposed that a cluster of synapses might be the smallest computing unit, because you only need something like, 5 to 8 spines activated together to generate a dendritic spike within a compartment. And if you consider this a computation, a nonlinear computation, which essentially that's what it is, right? You're crossing a threshold, you're mimicking the sigmoidal nonlinearity of the cell body, then you can think of having many such clusters of synapses within a long dendrite, and each of them would be a computing unit.
All right, I'm going to go real broad here. Where are we? I mean, you mentioned earlier already that we don't know we don't know everything about dendrites. Where are we in that progress? I mean, we have Hodgkin-Huxley models for neurons, right, and conductances and membrane capacities, et cetera. But in terms of, so I guess this is like two wrapped up questions. One is just where are we in the broad scope of understanding the properties of dendrites and how they contribute to neuronal function and cognition. And now I'm forgetting the second question, so we'll stick with the first.
Where are we? Okay. So in terms of tools for modeling dendrites, we use the same tools as we use to model the cell body. So Hodgkin and Huxley equations are perfectly fine for describing the different ionic conductances that are found in the dendrites. Capacitors also describe their membrane properties. But in terms of experimental data, we don't know as much. And unfortunately, people are now kind of abandoning the methods that would tell us more about dendrites, which would be patch clamping in vitro. So we're kind of moving away from that technology because it's not as sexy, and people are doing a lot of in vivo experiments now.
Yeah.
And in vivo, you cannot really map out the type of conductances that you have, at different parts of the dendrites, and you cannot measure their concentration, their conductances. So we know mostly about dendrites from studies that were done in previous years. And that's a shame. And we know very little about interneurons. We know more about pyramidal neurons, but we don't know a lot about interneurons, and interneurons come in such a large variety, of shapes and electrophysiology profiles. So when we want to study interneurons, the dendrites of interneurons, that problem becomes even bigger. Yeah. So I am hoping someone who will listen to this podcast will want to go back and study dendrites using techniques that can tell us about their electrophysiological profiles.
Oh, that's going to be a hard sell.
Yeah, I know. Maybe we will do it.
Yeah, you have to do it, right? And you are kind of starting to do it. I don't know. Are you patch clamping?
We do have a patch clamp facility here in the University of Crete that, in fact, my lab has helped establish many years ago. But in the new direction of the lab that we are starting now, we are also turning to in vivo experiments.
Okay, yeah. All right. So again, we'll come back to that. But it's overwhelming. So, okay, the neuron, right, which used to be considered the functional unit of the brain. We have lots of neurons, and they're all connected and recurrently connected and in complex ways. And you can, kind of think about the small circuit level in like brain areas. And that's kind of more coarse graining things. And you think, well, okay, I'm getting a little bit closer to like cognition, whatever that means. Mm-hmm. But it's already overwhelming to sort of think about the number of neurons and the way that they're connected. Then to think about not just the number of neurons, but, there are like, I don't know how many different types of neurons there are these days. What's the classification? Hundreds? Probably.
I remember something like 30 interneuron types at least, and these are the main classes. Yeah. And you have all the subtypes. I don't know how many different pyramidal neurons we have by now. Yes, right. Hundreds, I would say.
Then how many different types of dendrites are there? Because you have
how
active
do you define
versus. Well, that's what I'm asking, right? So the complexity is just off the charts, right? Because I wanted to ask you about what an active dendrite is versus a passive. Maybe you can answer that real quick.
Yeah, I can. An active dendrite is one that is able of generating dendritic spikes. A passive one is one that only integrates the signals without ever crossing a threshold for a dendritic spike, and therefore the integration is, sublinear, linear to sublinear, whereas the other type is supralinear in some cases, and then it can get more complicated than that.
Yeah, you're almost wincing right now. I mean, and then you have, just the different distance from the cell body, the different shapes, the different connectivities between the neurons, where they, between the dendrites, where they join and where they branch, et cetera. You mentioned synaptic clustering earlier, and we're going to talk about plasticity because then you have 100 different plasticity rules.
Yeah.
It just seems, do you ever just sit back and think this is too daunting?
Yes, and I often think whether it doesn't really matter that much, in the sense that in the light of evolution, everything that worked would get selected, and maybe we are placing too much attention on things that are different, but they're different simply because they all subserve the same function. So I often wonder whether this huge variability, this complexity, is not necessarily linked to a unique function, but rather there is a very high level of redundancy in the brain that, was never punished, evolutionary speaking, because it did not have a negative impact on the brain function. If that's even remotely true, it would explain why we have such a huge variability in the brain and why we don't really need to explain every piece of it, because maybe it's doing the same thing. Hopefully.
But you mentioned earlier you think algorithmically, and algorithms are very clean. And
not evolutionary algorithms.
Oh, well, explain that to me then, because I think of mathematical algorithms are clean. And it's almost like modern computational neuroscience sees like an algorithm, like let's say the backpropagation algorithm, right? Just to throw one out there, sees that as like somehow evolution is driving toward that. But it seems much messier to me so evolution doesn't work that way. It's not normative.
I don't agree that evolution is driving towards that. Evolution is definitely selecting the optimal solution, but it's not actively searching for it, which is what the backprop algorithm is doing. So in an evolutionary algorithm is essentially randomly making changes like mutations in genes. That's how they work, right? They randomly happen. And then those that lead to some advantage, they improve the fitness of the organism, they get selected. And the others slowly die off. So we get better by chance. We don't get better by, instruction, like the backprop algorithm, which essentially finds out how much away you are from your optimum target. You correct yourself to the right way so that you get there faster. That's another kind of optimization that I don't think personally is what the brain is doing, and it's definitely not what evolution has been doing for the previous eons.
So forgive me, I'm going to probably ask in a naive way. I guess what I'm sort of driving at is how should I think of algorithms, for instance, and mathematical, like an equation, right, that we want to map onto how the neurons and dendrites and circuits are functioning. Do I think of it as like an attractor sink, that it's just kind of, I guess via gradient, circling around, like some sort of stable or unstable attractors? Like how should I think about the mathematical tools that we use to map onto our messy biological brains?
First of all, there are mathematical algorithms for implementing, evolutionary algorithms, which is what I described, random changes, and then you just keep the good solutions. And the way you imagine those, at least in my mind, is not like a attractor that continuously, travels towards the minimum point, which would be the center, but it's like having multiple wells on a surface, and you try to move to the optimal, but meanwhile you fall into many of those local minima. And then there is something that pushes you out of this well. The energy goes up, and then you jump out of it and you fall to another one. And then slowly you converge, if you ever converge, to the right minimum. There is an algorithm called simulated annealing, for example, which is doing precisely this. Now, if we want to go back to neurons, the way that we learn is dictated by the different plasticity rules, as you mentioned initially. Now, these plasticity rules, there are so many and they're very different, and they don't necessarily simulate something like this, right? Maybe they simulate a part of an algorithm that when you look at it from a higher perspective will look like an evolutionary change. And maybe we have different plasticity rules because we want to attend to different aspects of learning. For example, if what I want to do, a young child, for example, doesn't really know much about the world, right? So the best thing to do is to attend to regularities, things that happen together, things that happen coincidentally. So how do you detect the regularity? Well, everything that fires together wires together, like Hebb said. So you have a local Hebbian rule, which is just looking for these things that consistently occur together. That would be one hypothesis for why you have a Hebbian rule, right? Mm-hmm. Let's see. And then the Hebbian rule is implemented in many different ways. You can have the spike timing dependent plasticity, which is very sensitive on the timing of these events happening together. we have the BCM, which is dependent on changes in the calcium. This is a slightly different time scale, but again, it's kind of a correlation-based algorithm. And you have, various of these kind of rules where neuromodulators come into play, but they all underlie the same idea. At least that's how I understand it, that we're looking for regularities in the environment. Yeah. And this is unsupervised, right? There is no teacher there telling the system that this is the right thing to do or not. So the teaching signal has to come from somewhere else.
Right.
Yeah. And in animals and humans, our brains are directly connected to our bodies. So we execute actions. And then the teaching signal comes from the result of the action, right? If you're trying to reach to take a glass and you fail to do it, then, it doesn't work. But if you succeed, then you have some kind of a signal that I got this thing right. So the teaching signal comes externally. It's the result of the action and not something that happened inside the brain, which somehow, of course, is propagated back to the brain and serves as a feedback, we think, feedback information. So it's a loop that involves, a thought, an execution, an end result, and a signal back. In artificial systems, what is the feedback?
Right. So it's just an external label and it gets back propagated. Yeah.
Which we give it as the answer to the fact that the artificial intelligence system does not have an arm to reach out and execute an action and generate the feedback.
Yeah. But I mean, okay, so let's go back to a biological system. So you have that feedback loop, but then not only do you have the Hebbian style plasticity, you have homeostatic plasticity, you have intrinsic plasticity, and these types of changes are constantly in flux. It's not like they're sitting there and waiting. Like homeostatic plasticity is just a function of the cells needing to survive and, self-regulating, et cetera. So how do we square all of those dynamic changes with some sort of, feedback that eventually it gets right, right enough?
So I think that all these plasticity rules, they are solving different problems and they act at multiple scales. Like homeostatic plasticity is needed to make sure that you don't keep changing the synaptic weights in a positive manner so that you reach a hyperexcitability and then you have seizures in the brain, right? So you have to scale the weights down to make sure that you don't burn your brain. You also have to scale weights up occasionally if there is something happening in the brain that is pushing towards depressing synapses because you want to maintain excitability. So as a result of learning, there will be changes in the synapses. And then to make sure that these changes are not driving the system away from a stability point, you have homeostatic plasticity. So they need to operate together with Hebbian plasticity, which is going to be upregulating or downregulating selectively some weights without a way to stop this thing, right? Because if you don't have a gradient descent, let's say, like system, then you don't know when you actually reach your target. It will keep on changing. And that's another one of the things that we're, frequently thinking when we're building models. How do the neuronal circuits know when to stop? What is the stopping criterion in the brain? Right. One stopping criterion could be that let's say you reach the limit of the resources that are available to the synapses. So you don't have any more plasticity related proteins. So you have to stop. Another criterion could be, that you've done so many changes and you run out of ATP, let's say. They will mostly be related to resource availability rather than a signal that says, okay, you got it right, now you need to stop. Because we don't really know what is it that tells a circuit, that now your task of updating is done.
Yeah. How do you choose what to implement in your models then?
Well, in fact, we are implementing lots of plasticity rules, all of them working together, and then we take one out, we take the other out, and see what is the impact on the model, on the model's ability to learn. And we try to figure out what is the contribution of each one of these rules. And I'm not sure if it's the right way to do it, but it's definitely a good way to gain insight as to the necessity and the complementarity of the different rules that operate.
What other way would there be to do it?
Well, many people just use one plasticity
Oh,
rule,
sure. Yeah.
and look at what you can get with a system that implements this learning rule. And I think that's a big problem because you miss out on the interactions between these different kinds of plasticity rules and the contributions that they have, let's say, at the circuit level versus a much bigger scale or a smaller scale.
All right. Let me ask you this. So there are, all these different types of plasticity, all these different types of dendrites, all these different types of neurons. I believe I read in one of your manuscripts that you suggested that the dendrite, should start considering the dendrite the functional unit of the brain. So I mentioned earlier that for a long time, the neuron was the functional unit of the brain. And now people are talking about, circuits, microcircuits as the functional unit of the brain and related to cognition. Does there need to be a functional unit of the brain? And might dendrites be that at least in your head?
So the people who first proposed, let's say, the dendrite should be the functional unit of the brain, they did so back in 2010, in fact. There was a nice review paper by Branco and Hauser, which was based on our work and their work and many people's work showing that dendrites are essentially doing nonlinear computations like the cell bodies.
Wait, I'm sorry to interrupt you, but I would be amiss without mentioning your work in 2003 showing that a single neuron is comparable to a two-layer artificial neural network. And since then there have, that number has grown, right? And so was yours the first study to show that?
To my knowledge, yes, we were the first to predict that essentially dendrites act like small cell bodies, and they can solve the same kind of problems using a similar activation function, a sigmoidal activation function, like the cell bodies of neurons. This was back in 2003. And then soon after, people started talking about having more such nonlinear computing units inside a neuron. So not only individual dendrites, but maybe you can break them down into their distal apical, proximal apical, and basal apical compartments. And in those compartments, you have multiple of these nonlinear units. So you have like a 3-layer, let's say, or a 4-layer neural network. And then recently in 2021, there was a nice paper by the lab of Miki Landon and Idag Segev, where they showed that you may even need a deep neural network of up to 7 layers to approximate the temporal computations that are done in a single cortical neuron.
Yeah. Okay.
So it's increasing. We started with a 2-layer pointing to a dendrites as a unit, and now it's becoming a multi-layer, a deep neural network in a single neuron.
Yeah. Okay. All right. I just wanted to mention that because you were talking about the 2010 review where the authors suggested that dendrites should be considered a functional unit of the brain. So carry on. Sorry to interrupt you.
Yeah. So I was saying that I think we are now beyond the individual dendrite being a computing unit, and the computing unit is much smaller. And I think that it's just a bunch of synapses, small clusters of synapses. Oh. That will be the computing unit of the brain, the computing unit of a neuron, let's say. Okay. Because you have units at multiple levels. As you said, a microcircuit could be the unit of cognition. So it depends on what kind of unit that we're talking about here.
Yeah, right. So you mentioned the synaptic clustering earlier, and I was going to ask you just to explain what that was and its significance, because you just brought it up again in terms of being a functional unit. So could you describe that more?
Yes. So this was another one of the early studies that we've done with Bartlett Met back in 2001, where we essentially showed that if you have dendrites that are capable of generating spikes, they are nonlinear dendrites, and you have a plasticity rule that depends on detecting inputs that fire together, and therefore they should wire together, so a Hebbian plasticity rule, but which rule is now local, so it depends on what happens in the dendrite, not what happens at the cell body. So if two inputs, let's say, fire together and they cause a dendritic spike, then they would undergo plasticity within the dendrite and they will become strengthened together. Mm-hmm. And as a result of such a rule, you have all the spines or the inputs that are correlated in terms of their firing profile to be co-strengthened within a given dendrite. And therefore, you form small clusters of synapses that are spatially close to each other because of their temporal, let's say, correlation. And temporal correlation in this case would also mean somewhat of a functional correlation, because typically inputs that project onto the same dendrite and fire at the same time they also carry similar information. If you think of the visual cortex, they are probably sampling from the same location in the visual field, let's say. Yeah. Or if you think of the auditory cortex and the animal is hearing a particular sound, these inputs would carry frequencies that are found in that particular sound, in that word, if you want. So the functional correlation would result in the spatial temporal strengthening of inputs in a small part of the dendrite. So that's which is mediated by this Hebbian rule, which is now local. So considers the ability of the dendrite to fire. As a result, now you have the formation of groups of synapses within the dendrites. They are close to each other. They are strengthened together, and they have the ability to fire the dendrite. We think that this is a unit because if a signal that comes in by 4 or 5 synapses carrying a significant enough information that has induced plasticity this way through this formation of a cluster of synapses, then this would be an independent computation that is propagated to the cell body through the dendritic spiking mechanism. So that's what we think is why we think is a computing unit.
A computing unit of a neuron.
You could think of it of a dendrite because you can have multiple such clusters within a dendrite.
Okay, because I want to, the dream, right, is to link these really low-level mechanisms, low-level, like a synaptic cluster, to higher-level cognition, and not just like say, well, piano playing is linked to these 3 clusters. That's naive, but have some sort of way to look at the different scales, right? And kind of link them.
Right.
And
I will answer that.
Oh.
Okay. Yes. So first of all, I should say that this clustering was a prediction back in 2001 when we used our models, and now it is verified experimentally in numerous studies.
Oh, cool.
People see that experimentally you have the formation of these clusters, and most of the times they carry correlated information. So, a prediction that was 20 years ago was verified, it started being verified 10 years ago, and now many papers come out. So I think that's really important for the role of models that I wanted to highlight. Now, how do we link this to a computation at a higher scale? So we have some really nice, I think, work with the lab of Alcino Silva, which was published in 2016, where we showed that the mechanism that links information across time, so like 2 memories that an animal experience that are separated by a few hours, is through the synaptic clusters. Because you have one memory, let's say a given context, that is formed in a population of neurons, and then a couple of hours later, the same animal experiences a different context, and now the axons that carry information about the second context end up being co-clustered to the axons that carry information from the first context. Why is that? Well, there are interacting plasticity rules here. One is the plasticity of intrinsic excitability, which says that a group of neurons that has learned something will remain excitable for several hours, and therefore it will be, let's say, ready to capture another memory. And this would be the mechanism that also underlies the formation of episodic memories in the brain. So that's how you link them. But beyond storing the memories in the same population of neurons, we predicted with our models that they would also be ending up forming clusters into common dendrites. I should say that this was a prediction of the model. And now we have a paper under review, which hopefully will come out soon, that supports, I wouldn't say 100% verifies, but supports this idea that this linking within dendrites is the mechanism by which you bind information across time. So that's how you go from a dendrite to a behavioral phenotype, at the circuit and the behavioral level, which is how do I take 2 independent entities, 2 memories, and associate them and link them across time.
So, I mean, I'm not trying to criticize or harp on this at all, but, the real dream is connecting a synaptic cluster to composing a symphony or imagining a story or some, like some quote unquote higher cognitive ability. The
way I think about it, but this is a hypothesis, is that these small clusters, we have a review on this, every cluster is like encoding a piece of a sound that you frequently hear together, like a word, right? Which consists of phonemes. Because these phonemes are frequently encountered together in a given word, they are encoded by a small cluster. So you can think of clusters as words or pieces of music. And then you can combine them in different ways by activating different dendrites, let's say, or dendritic compartments that contain these words and generate sentences and generate sounds or music or a symphony.
Okay.
So this gives you the opportunity to form many, many different combinations of items that have a meaning. They form a word or a sound. And that's how it's easy to encode this, using few resources like 5 synapses, but in a way that is, when you activate them, the signal will go through because it will generate a dendritic spike. So it's a strong enough input, let's say. And it's a way to increase the signal to noise ratio also by having these small clusters carry the meaningful information. That's how I think about it. But as I said, it's a hypothesis.
I mean, thinking just about music, and you said phonemes. I had David Popel on a long time ago, and I've had other people also, even recently, Earl Miller, and they're interested in oscillations and their contributions to, in David's case, like words and the different bits of words, like the phonemes and the whole words, etc., and just the rhythm that we speak and understand, etc. Do you consider waves as important? Have you thought about that as part of the story with an interaction with the synaptic clustering?
To be honest, I haven't thought about that a lot. We haven't modeled the waves in our models at all. I think they are important for sure, especially when you think about the role of interneurons and how they are modulated by, oscillatory rhythms. But I don't have much to say because we haven't really worked on that yet.
Yeah, but you do incorporate interneurons into your models.
We do, but we don't really study rhythms that much. We have them being modulated by rhythms, like in their input. So they fire at the peak or the trough of theta, depending on the experimental data. So we do that, but we haven't really studied explicitly what would be the impact, let's say, of these rhythms on encoding, which would be a very interesting question to ask.
Yeah, we are kind of enforcing them in some way then. But
yes, exactly. We're forcing them. They are not, an emergent property of the system.
Right. Yeah, you define the timing. The other thing I was going to ask about, when you were talking about intrinsic plasticity, so this has to do like with your work with Silva, the Engram. So you buy into the idea of an Engram that with intrinsic plasticity, right? You have correlated signals coming into a neuron, and it stays active or ready to be active in a state for a short period of time, right? So then you can associate more things, and that would be an Engram cell, correct?
So an Engram cell, by definition, is my definition at least, is a neuron which is involved in the encoding of a particular memory. That's why you call it an engram. So you learn something, and this something is somehow stored in the brain. It's, somewhere, and this somewhere is, a group of neurons that when you try to recall that particular memory, they become active. So that's an engram cell. It's a part of a memory. It's a cellular correlate of the memory. And engram cells, there is a lot of literature saying that they remain active for a few hours so that they can capture more memories. And they remain active because they have increased levels of CREP phosphorylation. So essentially, it's like lowering the threshold for somatic activity. So it's easier that they get activated by subsequent inputs. So yeah, I think the engram is a valid story. Many people are working on memory engrams. How else would a memory be stored in the brain? It has to be somewhere in the neurons, right?
Well, that's what I was going to take us to next. And I'm sorry, maybe I'm getting us too far afield, but someone like Randy Gallistel would say, given, this just constant turnover of plasticity, connections, disconnections, new formations, strengthening, weakening, homeostatic, intrinsic, all that's too changing, and you need something much more stable. And, his idea is that you have to encode it in some sort of longer form material like RNA or DNA or something, right? And I guess the story would be, that during this plastic time, this intrinsic excitability time for a few hours, it's actually the neuron, like subcellular machinery, quote unquote, molecules, right, encoding that activity in some sort of RNA sequence. Does that? I'm not sure if you're familiar with that story, but his argument is that it's just you can't. An engram is too plastic. It's changing too much. Like, how could a memory be stored in an engram?
Okay, so there are two answers to this question. First of all, I agree that there is a lot of dynamic activity in the brain. And in fact, this turnover is extremely useful, at least in our models and in animal models. It shows that if you have higher spine turnover, then you learn faster. One reason could be because you're overriding previous stuff, right? And that's why you learn faster. It is possible. But on the other hand, the number of synapses that are activated every time we try to recall something or we learn something is very small. It's like 2% of the inputs in a given neuron. So I don't think that capacity is really a problem here. Every pyramidal neuron has 10,000 excitatory inputs. If you only use your 2% of those for a given memory, and then consider that you have all those combinations of neurons that you can use to form a cellular engram, the number is huge. And even if you consider what we are claiming, that the actual engram is at the dendrites level and not necessarily the neuronal level, then it decreases massively. So I'm not sure I'm convinced with the argument that we run out of space in terms of storing memories. That's one thing. The other thing is that you have much higher, let's say, turnover or dynamic activity in particular particular areas of the brain, like hippocampus, for example.
Yeah.
Where its job is not to store these memories forever. Its job is, let's say, to create episodes. Its job is to link things together. And then, according to the two-stage memory hypothesis, whatever is really useful information is extracted and stored elsewhere in the cortex, where the turnover is slower. And the hippocampus is there essentially to reach out to these places and bring back the pieces of information that will then be integrated and form a memory. So there are solutions to this problem by existing theories, and I don't think they are unrealistic. Now, it is also possible that some aspects of, if you want to call it memory, are also stored in the DNA. People talk about epigenetics, as another mechanism for storing memories for a very long time. And I am not rejecting that idea. I just think that those are different kinds of memories than the ones that we use on a daily basis.
Okay. Yeah. So this is another thing I was going to ask you. So I'll just ask it now. Sorry to kind of interrupt there, but yeah, I mean, should we think about memory as being, at multi-scales also, right? And that
absolutely,
So you're familiar
and
with
distributed.
And distributed, yeah.
Yes.
Because you're familiar with the RNA transfer in worms and sea slugs? Where you can train a conditioned response in one organism and then extract some RNA, put it in another organism, and that new organism will have that quote unquote memory, which is a behavioral output in this case.
Yes,
yeah,
yes.
But so then you think, well, maybe that's not that complex of a memory. Maybe it's just that is somehow encoding the right activities for that given behavior or something. So is that how you think of it, as kind of a multi-scale memory?
Yes. Yeah. I think there are also experiments in mice when they've shown that there is a hereditary aspect of different behaviors that they think they are, transmitted, let's say, to the offsprings through epigenetic mechanisms in the sperms, I think, or both of the eggs. Can't remember it quite. And seen in multiple generations later, which are changes in the behavior. So that's why I'm saying I'm not rejecting that idea. I'm not fully convinced either, because you have a lot of behavioral mimicry here. So I don't know if you can for sure, make the distinction between an animal learning from another animal through behavioral mimicry versus, inherited memories. But I like to be open-minded. Let's say, and definitely I believe that memory is multi-scale and distributed, and there are different kinds of it taking place, different parts of the brain and possibly other organs.
It's fascinating. I find myself sort of surprisingly to me, just becoming more and more enamored with the multi-scale capacity, just the capacity, right? As for that to be such a beautiful, it's becoming more and more beautiful to me as we go along. Anyway, it's nice to still remain in awe of things, right? After such a long time studying stuff. You have children, right?
Yes, 3.
3. Oh, okay.
Yep.
Oh man, that's like 3 too many for me. But I have 2 kids. But do you ever worry, like, are you like me, that your former self wasn't as careful and not the best human? And that I worry, what did I pass on? Did I pass on some trauma through epigenetic mechanisms to my children? Do you ever think about that?
Yes, I do. Especially because in our line of work, we work very long hours. We're very stressed, and it's possible. I mean, there are studies showing that the stress, can impact the embryo in many unforeseen ways. So yeah, I hope not, but
maybe just the good parts. Hopefully,
let us hope.
I keep telling myself that. So again, I'm kind of jumping around here, but one of the things that dendrites do is they had this nonlinear effect, right? Which is kind of emulated in sigmoidal functions somewhat in artificial networks. But these days, artificial networks don't care about sigmoid functions. It's all ReLU, which are these like linear functions, and they do just fine. What does that mean? What should we take from that?
Very interesting point. In fact, the ReLU is not, most of the people, they don't use a fully linear one, right? It's saturated even. So it's not linear, first of all. It's not sigmoidal.
It's not sigmoidal. It's not logarithmic. Yeah.
It's not sigmoidal, but you couldn't call it linear, right?
It's linear with 2 parts. I mean, how picky do we need to be?
Well, a sigmoid any function can be approximated with small linear parts, if you think about it that way.
Okay, true. Your
so
calculus
it depends
is coming
how
through.
many linear parts do you need, right? That's the big question here.
Okay.
Yeah. Good question. They're doing great with ReLUs, I know. And we are using it in our networks as well. Maybe it has to do with the plasticity rules, the learning rules that these networks use, which are not very similar to the biological ones.
Well, so would the suggestion then be that those artificial learning rules are more powerful than the biological ones?
Well, I think that nobody can say that backprop is not a super powerful learning rule, right?
Well, people talk about how ungodly slow it is.
Backprop?
Because you're just taking very small nudging steps, right, toward a gradient, right? And it takes a machine.
Yeah, but you're always taking the right steps. You're always moving towards the right direction. So in fact, Heppian rules are slower.
Well, there's a guarantee, but computationally slow, perhaps. I'm not an expert enough to argue about this, but that's what.
Yeah, no, I don't think that the argument is that it's very slow. The biggest argument for us, at least in neuroscientists, is that I am not convinced that it's biologically plausible, personally. And because I don't see how exactly would, one layer know what the synapse of a layer, 3, let's say 3 steps deeper, how would that know a particular weight of a neuron? How did it contribute to the output that was computed 5 layers higher?
Right. Yeah, I mean, it's clearly not biologically plausible as such, but I mean, you're aware of the approximations, right?
I am.
Many of the approximations.
Yes, I am. That's one thing that I'm not convinced of, and the other thing that I don't really buy with this theory of correction is that a lot of the studies show that dendrites, in fact, compute associations. They compute coincidences. That's when you have the generation of dendritic spikes. It's not when you're making an error and therefore you don't have input coming in, but when you have 2 inputs that happen to coincide, so you are positively detecting conjunctive signals. Whereas in a method that is based on errors in order to correct a system, it doesn't really align well with the experimental evidence that dendrites normally fire when you have conjunctive inputs coming in. So these are the 2 things that, for me, they don't sit very well with the way dendrites are teaching the brain, are helping the brain to learn.
Is that why you're modeling spiking neural networks?
Yeah, we're modeling all kinds of networks, biophysical spiking and the artificial neural networks. And I think the spiking neural networks are important for many reasons. First, they're closer to the real thing, right?
Oh, I thought you were going to say efficiency.
No, I was going to say they can generate spikes.
Okay.
So they're closer to biological neurons.
Okay.
Secondly, they are much easier to be implemented on neuromorphic hardware, which is designed for this purpose. And I think that neuromorphic hardware and any new technology that is trying to consume less energy is the future. So yes, they are efficient in that sense. They are faster. You can implement unsupervised learning rules much more easily on these systems. And you don't have to incorporate all of the complexity of the biological neuron like we did with Hutchinson and Huxley models that we also use in the lab extensively. So I think they're a very good compromise between the detailed networks and the very, very abstract networks that we use in artificial neural systems.
How do you decide what level of biological detail is the right level? Is
Excellent
it a matter
question.
of your computing resources instead? I'll just leave it as an open question instead of putting answers in your mouth.
We try to follow Occam's razor, so as simple as possible, but not simpler to answer the particular question at hand. So if you want to study receptors, let's say how the NMDA receptor or how sodium channels influence dendritic integration, I would go to a detailed biophysical model because that's the only way to capture the kinetics and the spatiotemporal interactions between mechanisms in a dendrite. You have to incorporate the morphology and the conductances of the other channels and all the gating parameters. So if you want to go very subcellular, you need a detailed model. But if you're interested, let's say, in how dendrites impact circuit computations, then you don't really need all this detail. You just can abstract these local computations into a mathematical function, a transfer function, like, the induction of a long-lasting spike, let's say, not a purely mathematical one like a sigmoid, but something that somehow incorporates the temporal dynamics of dendrites and put it in a spiking neural network. And then now you can build a big network because you don't have very expensive subunits now. You don't have tons of differential equations. You have a few. And then simulate large circuits and see how dendrites influence circuit computations. And then if you want to go to machine learning and see whether you can make a difference there by adding, let's say, some of the dendritic features, you can also build classical artificial neural networks, that don't consider time like spiking neural networks. So we do all three, let's say, approaches in the lab.
So this is what, maybe you can just say a word about Dendrify.
Dendrify. So Dendrify is a new tool that we just published in January. It was published in Nature Communications. It's a tool that is very close to my heart because the idea is to convince the community that you can put dendrites into your model in a very, very easy and straightforward manner. So essentially, we find a way to describe dendritic nonlinearities without using the Hutchinson and Huxley equations, which are computationally heavy, but also, mathematically demanding for some people, right? So we are describing the nonlinearities with event-based mathematics. So it's very simple. You have an upswing and a downswing, let's say, current to simulate an action potential, a sodium action potential in the dendrite. And similarly, for NMDA and calcium spikes, we're now building in those aspects. In fact, initially they were not there. So this is a tool. It's very well documented. It's built on Python. We have examples in there for any naive user so that they can go in and play. And we are maintaining the tool in the lab. And we are going to give a technical workshop in the CNS meeting this summer. So if people are interested, they can attend that. And we're looking forward to convincing more people to work on dendrites.
How's that going? The convincing?
I think it's going very well so far. Yeah. I see a lot of publications, in fact, lately incorporating dendrites, not necessarily through Dendrify because this was just developed, right? But a lot of people are getting more and more interested about dendrites, and especially with respect to their impact on the machine learning and AI field, which is great.
Yeah, well, I don't know. Is it? I mean,
it is, of course.
Do you think that now I want to ask about the AI field. So, I mean, I don't know how much interaction you have with people in AI in like the industry side, right? Applications and stuff, but
Not much, I have to say.
So they don't give a damn about dendrites, I'm sure.
Yeah, I know.
Someone I can't remember who it was talking about, it seems like almost every lab, especially computationally oriented labs, considers like building, well, we'll build this standard tool and make it open source, and then the community will use it, and we'll share it. And it takes a lot of effort to build something like Dendrify. And then often what happens is you build the tool, you put it out there, and then you're expecting like a lot of collaboration and to have interactions and stuff, but you almost have to like sell it to the community, right? How much of what you're doing, by the way, how much of what you're doing is actually, getting out there and saying, use Dendrify, here's the thing, or people coming to you?
I don't do much selling, to be honest. I know I haven't done it for two reasons. First of all, I think we have to convince people to be interested in dendrites and then to use Dendrify, right? Because the dendrites community is not that large anyway. Yeah. So I'm definitely focusing on convincing people that dendrites are important and they should be looking into them. Dendrify essentially, we didn't really initially wanted to make a tool. It's the work of a PhD student that just wanted to make his life easier. Because he wanted to build the spiking models, and he wanted to avoid complicated equations, so he came up with Dendrify. So we did not plan to generate a tool for the community, and maybe that's why we're not selling it as much. But there are a lot of people that expressed an interest so far to use it, and they've reached out and we've helped them. And I definitely believe that it's not enough to make a tool and just put it on a website, because people need to be trained to use it, right? So you have to offer some training opportunity. And in fact, it's a responsibility. If you want the community to, take advantage of what you created, you have to show them how, right? And that's why we are organizing some training workshops for that. But we don't plan to become tool developers. We just want, whatever we think is helpful for us to make it available to the community who is interested in dendrites.
But now you can't help it, but you feel that responsibility. It's taken on a larger role than originally planned, just to solve one student's problem.
Yeah, that's a good point. I'm hoping the student will still be interested in maintaining it.
Running a lab. I'll never know.
Yeah.
Perhaps.
Maybe you're better off.
Oh, I don't know. What do you think? You think I'm better off? Now, I'm kind of like looking for a job. I don't think I'll be running a lab necessarily, but well, maybe that's a separate conversation. Running a lab, yay or nay?
Running a lab is difficult, especially if you are in Greece. Funding resources are very limited. We already mentioned the lack of collaborations and, local community.
Dendrites, which there are like 7 other people in the world studying.
Yes. Yeah. I mean, that may be good because it offers a niche opportunity, but it doesn't, make sure that you have enough funding for it. So, the efforts to secure funding are exhausting. I think they're exhausting for everyone nowadays, and that's why many people leave academia, unfortunately. But yeah, it's a lot of work and a lot of responsibility towards the people in the lab, right, that depend on you securing funding for their future. So on the other hand, it's super fun and it's so rewarding because we're always trying to figure out something new. I mean, I love my job, right? I wouldn't change it for anything, but it's certainly very stressful.
So I just moved to Pittsburgh, Pennsylvania, where I did my graduate school. And there are a couple friends still here. And I reached out to them saying, hey, it'd be great to go, grab a coffee or a dinner sometime. And they're like, yes, great. But it needs to be like 2 months from now, because they're running on, quote unquote, running on fumes, and I don't think that never changes, does it?
Well, I think maybe the US is worse than Europe. But yeah, if you're running a successful lab, or you want to have a successful lab, it means working overtime, working too much. Yeah. And, I often wonder whether it's worth it because there are other important things in life, family, health, enjoying life.
And you've already ruined your children by passing on your stress.
Well, they don't want to be scientists. So
good for them. Good for them.
Good for them. Yeah.
Let's talk a few more minutes about, all right, so you've created this Dendrify, and it went into Nature Communications, which was that a hard sell getting it?
It was easier than I expected.
Oh, really? I don't know. Like, I haven't like watched, and I don't have a track record, but like it's essentially a resource, and
it's a tool. Yes,
it's a tool. And maybe that's what Nature Communications specializes in. And I'm just naive.
No, it doesn't. I think the reason they really liked it is because it bridges multiple fields. So it's good for modeling in neuroscience, but you can also use it for machine learning and artificial intelligence applications, and it's readily implementable in neuromorphic computing. And we also, with it, we proposed a framework about why dendrites are important and how you study it. So it was a bigger thing that spans multiple communities. And I think that's why they really liked it.
Okay.
Yeah.
Okay. So way back when, I had Nathaniel Daw on my podcast, and he was talking. So he is traditionally a computational cognitive scientist, neuroscientist, and we got into a conversation about. I'm sure you do, through NeurMatch and everything. We got into a conversation about how he and his friends, lab advisor friends, principal investigator friends, were thinking hard about whether they should start an experimental lab, because they have all these theoretically driven computational research approaches, and that they make predictions, but then you have to like convince people to do the experiments. And the question is, well, should we just start our own wet lab to like do the experiments that our computations suggest, our theories suggest? And I said no, but you didn't take that advice, because you have transitioned your lab to become also an experimental lab. How has that transition gone? Was that a wise move?
Well, it's kind of early to tell, because we haven't published our first experimental paper yet. Until that moment comes, I don't think I can answer this question, but I can tell you why I did it. And I did it because, well, one reason is, as you said, we have to convince our collaborators to do the experiments we want, and that rarely happens. In fact, most of the time, and I am a strong believer of collaboration. I have a lot of collaborations with many people, but most of the times it's, the model comes in to explain the experimental data, which are generated by the experimental labs.
Right.
And I really wanted to be able to have a control of the type of experiments that we do, so that we can test our own predictions. As you said, it's let's say the satisfaction that you get by making sure that someone looks into all this hard work that you've done over years to generate these testable predictions. The other reason is because, the animal is the real thing. The model is a model. It's great. It's an amazing tool, right? It can allow you to do experiments which are not even feasible experimentally. So it has an immense amount of power. But the real thing is what happens in a biological brain. And I really wanted to get a better insight, of these processes. It's been fun. It's much slower than I expected, more expensive than I expected.
The development, or the actual research, or like getting the
both
labs set up, and both.
Yeah. Both. I mean, in behavioral experiments that we do right now, they last a month. And then you have to wait another 2 months for the animals to be at the right age, and then start again. And it's taking forever, which is something I had not really considered, because modeling is much faster.
Thank you for saying that. I've been, anytime I ask that, people say, well, no, it's not necessarily faster. It really is faster, right?
It is faster.
Yeah.
It is faster. I mean, it can take a very long time to troubleshoot, to find a bug, to optimize a model, but it's faster. It's definitely faster. It's not significantly easier. I wouldn't say that. In fact, I think that doing experiments is easier because you need to have really good skills with your hands. Whereas for modeling, you really need good skills with your brain.
And with lab experiments, you're doing a lot of waiting around often.
Yes,
also.
Yeah, exactly. Which is nice. It's, more social, more relaxing. It gives you more time to think.
Yeah.
But producing results is faster with models than with experiments, for sure.
So when can we expect the first experimental paper?
We're not very far. But then again, I don't have a good feeling of what reviewers will ask, right?
Right.
Because you have to submit it first and then see,
Yeah.
And it's also always the fear that when you are a newcomer in the field, you have to convince people that you can actually do experiments. So Yeah.
Well, good
We'll
luck.
see.
Good luck
Thanks.
with that. These are rodents, though, right?
Yes, we're
Yeah.
working with mice, and we're doing in vivo head fix behavior, looking at the prefrontal cortex and looking at behavioral flexibility and how the dendrites or spines and spine turnover may contribute to behavioral flexibility. And in fact, we started this project with funding from Germany together with Matthew Larkum. So we have an aspect of the work that is done in Matthew's lab, actually, funded by a German grant, and now we're continuing it here at home at IMBB.
Yeah,
and I'm actually very happy because we have set up the first 2 photon microscope in the institute.
Congratulations.
Yeah. So this is going to help others as well.
I think it helps create excitement around dendrites that people like you and like Matthew are working on them and doing such interesting work. I mean, you're good spokespeople for the dendrites community, maybe, I would think.
Thanks. Yes, we'll try our best.
I asked about the rodents because ultimately we're interested in human brains, right? So first of all, you start many of your talks with a slide that shows the progression of dendrites over the course of our lifetime. Yeah. And it's pretty striking, actually. You and I are right now at the peak of our dendritic arborization in our lifetime.
You are.
Well, I'm at my peak, but that ain't saying much for me. But it really shows. It's pretty striking because, from birth, you have these neurons with kind of small branches, right? At least on these slides that you show.
Yeah.
And then from, I think, 30 to 60, they get bigger and bigger. And then, there are these beautiful outstretching trees when your age. And then we're going to start to decline, sooner rather than later, I suppose. And then they kind of shrink back down again. And you show dementia and Alzheimer's and how they shrink back down in different conditions and just with aging. But, and those are human cells that you show, right?
Yes, those are human cells.
I'm not sure about the difference between human aging and other organisms. For example, mice. I'm sure that you know the answer to this, like the dendritic arborization, along
Actually,
there.
I don't. I should look it up and have a similar slide for mice. I bet it's similar, but I have not looked at the same figure for mice across.
Their cognitive peak must be
around 30 days.
30 days? Months. I was going to say 2 months.
From 30 days to 2 months, they are adults. And I think that's when they have their, up to 6 months, they're still considered young. And then they start to age. But I have not seen a respective slide with, the dendritic anatomy for these animals. So I should be better prepared. I'll find one for my next talk.
Yeah, please prepare yourself better. This is ridiculous.
Yeah. Yes.
But are human dendrites special?
Excellent question. Yes. Well, some people think so. I mean, we even thought so 3 years ago when we published this paper with Matthew Larkum in Science, where we discovered that there was a new type of a dendritic spike in human dendrites that was not previously seen in rodents, and which allowed these dendrites to solve complex mathematical problems like the exclusive OR. But then a year later, the same kind of dendritic spikes were found in rats.
Oh, yeah.
Yeah. So at least that particular aspect was not what makes us humans, was not what differentiates the dendrites of rodents from the dendrites of mice. It's a very interesting feature, nevertheless, because it was the first time that we discovered a mechanism that has a non-monotonic activation function. As we discussed previously, we use sigmoid activation functions, which are monotonic, to model dendrites in abstract models and in detailed models as well. And this was non-monotonic, so it goes up and then it falls, which means, and that's why it allows, the dendrite to solve the exclusive OR problem, which requires a non-monotonic function. So it's interesting to know that there are these kind of computations in dendrites as well, even in rodents. We had not considered that before. But whether there is something else that is unique, I mean, we know there are differences, right? There are differences in the size, there are differences in the compartmentalization. Human dendrites are much more compartmentalized than mice neurons.
What do you mean compartmentalized? Like smaller, like more
So they are longer. First of all, they are bigger.
Yeah.
And the compartments, they don't communicate with the cell body with the same strength as they do in a mouse neuron. So there is much more attenuation of the signal to reach the cell body, which means that they are more independent because they don't talk to each other as much as they do in the mouse, and they don't talk to the cell body as much. So you can think of them as having more parallel units if we are to think each part of the dendrite as an independent unit, which could be one reason why humans can do more difficult cognitive tasks if you correlate the number of parallel units in the mouse and the human. That could be, one reasoning, let's say.
Yeah, I guess we could tell ourselves a bunch of gesso stories about this, though, right? So I immediately thought, well, it's more compartmentalized, more attenuation. That means like you can have like subtler differences and finer scaled and thus higher capacity. But I'm just telling myself a story there, perhaps.
Well, I mean, we know that if you have more compartmentalization and every compartment is nonlinear, then you do have a higher capacity. I mean, we've known that for mathematical models. So it's not the story. I think you can, it's a convincing story, let's say. Okay. Whether that is the reason we can solve more difficult problems, though, that's, hypothesis.
Yeah.
Because you have many such neurons and circuits in the mouse brain that maybe is sufficient to solve the same type of problems that you would with fewer neurons in the human brain. So we cannot tell that this is a differentiating factor. It's a matter of scaling up. If it's just the number of compartments, it's just a scale up version. And is that the reason why we are better? Is it a matter of scaling up?
We also have like a larger brain to body ratio. We also have opposable thumbs, which helps. Right.
Yeah.
Somewhat. Has your career gone, let's see, have you made the right choices? Did you get in the right field? Are you doing the right thing? Are you happy with the trajectory and the journey?
Yeah, I think I've done a lot of wrong choices that ended up putting me in the right spot.
Oh, that's a nice way to put it.
Yeah. I mean, I'm very happy with where I am right now and with my life and career right now. But, the path was not easy. As I said, I couldn't study neuroscience early on. Then I went back somehow. Then I returned to Greece and there was no computational neuroscience. I did bioinformatics for 10 years. So I had to change my research direction and then wait for an opportunity to come back to neuroscience. It was complicated. Yeah.
Luckily, your brain is very plastic with all those dendrites.
Exactly. I think I feel fortunate that I was able to, let's say, take advantage of all of the misfortune, if you want to call them like that, or opportunities to deal with different things. So I ended up being where I wanted, even though the road was not the easiest one.
Yeah.
The path was not the easiest one.
Okay, Yiota, I have one more question for you. And I don't know if you can answer this, but if you kind of zoom way out and just think about how you have viewed dendrites with respect to their importance and role in computation and life and intelligence, how have your views changed over time? How have they evolved?
Well, in fact, they have, because initially I was thinking that dendrites are really, really important because they allow the brain to do more advanced, let's say, computations that we could do with the point neuron systems, which is true, right? I mean, a point neuron cannot solve a nonlinear problem, whereas a neuron with dendrites can, and we've shown that, right? But I now kind of think that their more important role is not necessarily their ability to solve more complex computational problems, but they do this as a means of increasing the efficiency and the power savings and the resource savings of the brain. And that you could do the same stuff with point neurons, but it will be so much more expensive. So now I look at them from a slightly different perspective, trying to figure out what are the advantages of that are offered by dendrites in circuits, in large neural network systems, that are not necessarily focused so much on the improved computational capabilities, because, we've known they are there, right? We've shown them. But with respect to efficiency, speed, energy, making a system more sustainable, and how we can take this knowledge and use it in other fields. Transfer the fields to machine learning and AI, for example.
So what I want to happen, I want to be more excited about efficiency, just as its own. I know it's important, but just from an intellectual curiosity vantage point, like I want to think, oh yeah, efficiency is really cool.
Okay, let me give you some examples.
Okay.
Edge devices, right? If you want edge devices, devices
edge computing.
So yes, edge computing. So if you want ChatGPT or the next version of it to run on your smartphone or even something smaller, you have to fit it in, right?
Yeah, I know that's important.
But that doesn't, you don't care about. Or let's say the sustainability of our planet. We cannot burn in
I know it's important. I know our planet is important. It's just there's something that's not like the wonder and awe of our intelligence is not contained. I know efficiency is part of the story, but I'm thinking more of the capacity and the plasticity and all of that stuff is like cool to me, right? And efficiency is necessary.
Yeah, I see what you mean. Also, but all of these properties, why are they more cool? Why are they cooler than doing all the kind of solving all the kind of problems that you're interested in and doing it in an efficient way? Because you need to have both, right? Efficiency on its own is not enough. You need to also have very powerful computing systems.
Mm-hmm. Right.
Right.
Well, not if you're super, super, super efficient, right? Then you can reduce the computational power if you can just run a lot faster and with less energy, because it would just do this. It'll take longer to compute, but it'll be compressed in a lower power efficient system. I don't know. I'm not a computing.
No. But what I'm saying is that we want to have smarter systems, right? We want ChatGPT to be better. We want the AI to reach the generally. Well, I'm not sure we do actually. Hinton resigned, right? So this is a controversy issue, let's say. But let's say if we wanted to have systems that learn like the human brain, they don't forget, they don't mess up things, they are great. And you run out of technologies to generate them because they are not efficient. You will never get there. So you need to have both. You need to have a way to build intelligent machines, and you need to have a way that these intelligence machines, can run on a laptop.
Here's why I'm not excited. Here's why it's not exciting to me, because what is exciting to me is the explanatory part, the understanding.
You're right. I should have mentioned that. That's a great point. So the other reason we are interested about understanding how dendrites achieve efficient computations is because we want to understand how they do it, right? Yeah. And in a computational model where you explicitly simulate dendrites and you look at the readout impact at the circuit level, for example, and you measure the differences with and without dendrites, and you try to look at what is it that dendrites encode if you have them, and what do you miss out if you don't have them, you increase the explainability of these systems, massively. So you no longer have a black box. That's another reason why we care a lot about understanding how dendrites contribute to, advanced computations.
Okay, you've taken me one step closer to caring about the
to caring. Otherwise, you wouldn't care about dendrites. That's terrible. That's really sad. You have to change that.
Yeah, no. I think it's exciting. I think it's really cool. As I started, this podcast, this episode. It's just daunting. It's like one step further removed from being able to grasp what's going on, right? Because there's so many more dendrites and so many different types of plasticity within the dendrites and configuration, and as you've demonstrated.
Daunting. Yeah, biology is complicated, right?
Yeah.
But we've come a long way, and now we know so much more than we knew 20 years ago. And now the technology has advanced so much that we can manipulate dendrites in vivo in the behaving animal. There are like 5 or 6 labs we can do it, but they can do it now, right? And we found really exciting things experimentally, that they underlie perception, that they may be responsible for for anesthesia. These studies are from the lab of Matthew Larkum. They may be implementing predictive coding across different regions. So we've learned a lot of stuff about why dendrites are important in the biological brain, right?
Well, if we can take some of this understanding and transfer it to artificial systems, that's already a major win, I think.
Yeah, but the AI community doesn't care.
Well, you're wrong on that. There is a lot of papers right now coming out talking about the need to come up with more sustainable technologies.
Sure.
And in the search of more sustainable technologies, I bet they will care. They are already care about bio-inspired properties that they want to add to their systems in the hopes of making them more efficient. Dendrites are unfortunately not on the map for them. And I think they're not on the map yet because they are thinking of them in terms of the computing capabilities which they offer to the systems as additional units, and not for their efficiency properties. And that's why I think it is important to understand them and characterize them, because they will become a key player in the years to come.
What are the things that they have their eye on in bio-inspired, like neuromodulation, modularity?
Neuromodulation, different plasticity rules, biological plasticity rules, restricted connectivity, sparsity, things like that. Some people are starting to look at dendrites. Some people have looked at the dendritic calcium action potentials that we published, so the human-like, let's say, features, but again, in hopes of providing more advanced computational power with these systems, which I really think that should not be the main focus for the bioinspiration.
All right, Yiota, I think that I've taken you far enough. I appreciate all your work in bringing dendrites to the fore as well, and I hope it continues to grow. And although you'll continue to stand out, especially now that you're going to be doing experiments to back up your modeling. So thanks for joining me here. I really appreciate it.
Thank you so much, Paul. I had a lot of fun. I hope people enjoy this.
I alone produce Brain Inspired. If you value this podcast, consider supporting it through Patreon to access full versions of all the episodes and to join our Discord community. Or if you want to learn more about the intersection of neuroscience and AI, consider signing up for my online course, Neuro AI, The Quest to Explain Intelligence. Go to braininspired.co to learn more. To get in touch with me, email paul@braininspired.co. You're hearing music by The New Year. Find them at thenewyear.net. Thank you for your support. See you next time.